Okay, welcome to Honors Biology or On-Level Biology, whichever class you're watching this for. This is going to be our first introductory video, and this video is on experimental design and scientific method. Now, I'm going to use an example to kind of teach you all of the parts of the scientific method. So, what if I wanted to know which of these French presses would keep my coffee warmer for a longer time? So that would be my testable question. And now I would want to come up with a hypothesis. And my hypothesis would be that this metal thermos container would keep my coffee warmer for longer than this glass container would. Now, the independent variable in this experiment would be the independent experiment would be which type of French press container I was using. Am I using the metal container or the glass container? So as a scientist, I control the independent variable. And in an experiment, there can always only be one independent variable. Because if you have more than one independent variable, then you don't know whether the outcome of your experiment is due to one variable or the other. So we always have only one independent variable. The dependent variable would be how long it would take for the coffee to reach a particular temperature. So I could set up this experiment and I would use boiling water from the same coffee pot, so we knew it was starting from the same temperature, put it in each of these carafes, and then keep a thermometer in there and see how long it took to reach a predetermined temperature that I decided upon in advance. So oftentimes, or always actually, the independent variable is the one the scientist decides in the beginning what those parameters are going to be. The dependent variable is the one that the scientist measures, and it is affected by the independent variable. So it's as a result of the independent variable. Now, the control group is not quite as clear in this particular sample of an experiment. I would say since this is the first French press that we started out with, and this is the newer French press that we got because we thought it was an improvement, that the data that we got from the glass container would be our control group. So a control group is always used as a method for comparison of your experimental data too. So the control group is your basis for comparison. So the other thing that we need to talk about is what is a controlled experiment versus a control group. Your control group is your group of data that is used as a basis for comparison. Is the data or is the experiment controlled is a totally different question. So if the experiment is controlled, all other factors would have to be the same except for the one factor you're testing. So in this case, we decided it was maybe the metal container, like the material that the French press is made out of. So I'm actually kind of glad that you can see that these are two different sizes because it shows you a flaw in my experiment if I did compare this French press to this French press. So we would want to make sure that the only thing that was different between my two French presses was the material, metal versus glass, if that's what our question was. Of course, as you can see, the volume of the liquid would make a difference. The surface area of the container compared to the volume would make a difference, so I couldn't really make this a controlled experiment. It would also matter that the lid were the same. These lids are not made of the same material either. So I really couldn't do a very good controlled experiment with these two particular French press. So the thing to consider is what is everything else that would have to be kept the same in order for you to only be testing one thing. And those other factors are sometimes referred to as controlled variables. I don't really like that term for two reasons. First of all, if they're the same, they're not really variables. Secondly, we have a controlled experiment and control group. I mean, I think it gets kind of confusing when we have too many terms that have the word controlled in it. So I think a better term to use for those other things that we would have to make sure were the same, I like to refer to them as constants. So in my experiment, our constants would have to be the size of the container, the volume, the surface area would have to be the same, the lid would have to be the same, the initial temperature of the water would have to be the same, and we would certainly want to use the same thermometer. Now, you're going to come across the terms qualitative and quantitative when you're talking about data. Quantitative data is any data that has numbers associated with it. Think quantity. So if you're counting something, that's quantitative data. If you're describing something like the color, the aroma, that is qualitative data. You don't always have to have quantitative data, but usually that's what is, considered more scientific. So the other thing that we need to talk about is sample size. Now, what is the difference between sample size and number of trials? It's very often confused by students. So in this experiment, I would have a sample size of 1. That would not really be a very sufficient sample size. But of course, I also only have 2 French presses at home. I'm not going to buy 30 French presses that are glass and 30 that are metal for the purpose of this experiment. And it's actually kind of good that I only have 1, because we can talk about how important sample size is. For example, if somebody was doing a drug trial and they had come up with a new type of medication and they tested it on 3 people and they said those 3 people got better and didn't have any adverse side effects, you probably wouldn't be convinced. But if they tested the new medication on 500,000 people, well, that's probably a little bit unrealistic, but let's say 5,000 people, and none of them had any adverse side effects and it was effective compared to a placebo pill, which they would have to use as your control group, then you might be more convinced. And in fact, the best kind of study is the double-blind study, in which the scientist doesn't even know which patients were getting the placebo pill versus the new experimental medication. So that doesn't apply to my example with my coffee makers, but it does apply to a lot of human studies with medications. And so I also want to talk about what is the difference between sample size and trials. So in this scenario, I would have a sample size of 1, because I have 1 of the experimental group and 1 of my control group. Not enough as for scientific study. But if I did the experiment multiple times to verify my results, that would be a new trial. So oftentimes when scientists are doing an experiment in a lab, they'll do the lab, they'll get the data, and they'll try again to see if they can get the same results. And in fact, it's actually even better if somebody from another lab does the same experiment to see if they can verify and get the same results. Students often get the idea of sample size confused with trials. So the number of trials is how many times that you do your entire experiment. It seems like a lot of time for school experiments, they tell you you're doing 3 trials when they really mean you have a sample size of 3. A sample size of 3 would not really hold up to scientific scrutiny, but when you're doing a science experiment in a classroom setting, oftentimes the teacher is limited on supplies and time and funding, which wouldn't be the case if you had a grant at an actual research institute. So that's why we tend to say, hey, 3 is a magic number if you're in school. But it's not really actually a great number. So that is the end of my scientific method introductory video.